Hello, biologists. This is going to be a brief lecture about the cell cycle and mitosis. So by the end of learning about the cell cycle and mitosis, the goal you can see below, we're going to determine the different stages of the cell cycle. We're going to draw the stages of mitosis. We're going to be able to explain what's going on in terms of the cell cycle and mitosis, and also be able to understand why the cell cycle is so important in animals. So, what is the cell cycle? So, the cell cycle is essentially a series of events that takes place in a cell before it splits into two cells. So, we're going to start with an original parent cell, and then by the end of the cell cycle and mitosis, we're going to have two identical daughter cells. So, why do cells divide? The first reason that they divide is because of the need to grow. So, growth. Um, we need to increase the number of cells in the body. And so we know and have learned that cells are limited in terms of their size. They can't get bigger and bigger and bigger and bigger because diffusion will take too long. The DNA will be inefficiently giving out information to make proteins for the cell. So we can't have this huge cell, so growth is one of the limits. We also need to be able to repair. So when you, for example, have a cut on your finger, you want to be able to replace those damaged cells. So this process of repair allows us to replace dead, damaged, or old cells. So just to kind of give you a scale here, you have about 1 billion cells die every hour, and that means that they have to be repaired and replaced. All right, the third reason is reproduction. So we can create new organisms asexually through cell division. And then, this wouldn't be necessarily for humans. We do have body cells that do division, but we wouldn't necessarily call that reproduction. But bacteria, for example, they're going to just split in half and divide, and that would be how they reproduce. All right. So the cell cycle is broken into two distinct parts. The first part is called interphase. And as you can see, it is the longest phase of the cell cycle. And its big job during interphase is to have the cell repair any damage. It's going to physically grow in size because it needs to prepare to divide. And in addition, the DNA is going to double, right? Because if we're going to have two new cells at the end of the whole cell cycle, we're going to need a set of DNA for each of those new cells. Then we have the M phase. So the M phase of the cell cycle, as you can see here, it's also called the mitotic phase for mitosis. And in the mitotic phase, the cell is going to divide. So you can see in the diagram that it starts with an initial cell, but by the end of the mitotic phase, you see there's now two cells. And the cells are going to divide into what we call two identical daughter cells. So let's talk a little bit more about interphase. So interphase, it is the longest stage of the cell cycle. It takes up about 90% of the cycle. So really the cell for the majority of its cell life is going to be in interphase. During interphase, it is preparing for division. It is getting ready to split in half, and it has to have a whole bunch of things set up and ready to go before the cell can enter mitosis and actually start the process of dividing. So we can break this interphase period into three major phases. The first phase is called the growth phase, and it's called G1. And in G1, essentially what we're going to do is we need a lot more cytoplasm, right? Because we're going to ultimately have two new cells. So we're going to need enough cytoplasm for two new cells. And we're also going to be making sure that the cell has enough nutrients to get ready to be two new unique cells. We then have the S phase of interphase. That stands for the S stands for synthesis, because in the S phase, we're going to be doing DNA synthesis. We're going to actually be creating a full new copy of the cell's DNA. like I mentioned, because at the end of mitosis, we're going to need two full sets of DNA in each cell. So this is the step where it makes a full new copy. Finally, we have the G2 phase. And this is when things get organized. This is when we are doing some final growth sort of stuff. We're really preparing to enter into that mitosis phase where we're going to start to actually do the splitting. So things are going to get organized. Certain organelles are going to be moving to the right places to prepare to split. Okay, so let's just quickly talk about something called chromosomes. Okay, so we've learned that inside of our cells, we have an organelle called the nucleus. And inside that nucleus, we have our DNA. Now, most of the time, that DNA is found kind of like if you were to look into the cell, it would look like a whole bunch of messy string. Okay, but there, we're going to see during a phase of mitosis, that DNA, instead of being all stringy, is going to condense. It's going to coil up on itself, and it's going to coil into these things called chromosomes. It's essentially just condensed DNA. And the whole reason for this is we want to get our DNA nice and tightly packaged before we enter into splitting up the cells so that there's no mistakes, so that none of the DNA gets broken. So instead, they're on these nice, tight little packages called chromosomes that are all bundled up. And it's a lot easier for cell division to take place accurately and efficiently. In terms of humans, we have 46 chromosomes in our cells, 23 from our mom and 23 from our dad. Okay. Oh, and sorry to fill in your notes here. So it is condensed DNA, and the whole point of it being in this chromosome form is it allows the DNA to move through mitosis without getting damaged. Okay, so now let's talk about the M phase or the mitotic phase. So this is actually the shortest part of the cell cycle or shortest stage of the cell cycle. It's going to be about 10% of the time the cell will be in the M phase. So what's happening here is this is when the cell is actively splitting. And in terms of how mitosis works, you're going to see that we have it broken down into 5 distinct steps where really unique and different things are happening at each step. So let's kind of break down each of these 5 phases called prophase, metaphase, anaphase, telophase, and cytokinesis. So as we can see here, in prophase, this is where we can start to actually see the chromosomes. They become visible. So as you can see, we can clearly and distinctly see these kind of like wormy looking things. Those are the chromosomes. And so before, you can see at the very beginning of prophase, it doesn't look like they are condensed yet. They're still sort of that big mess. But by the end of prophase, you can see that they are in those nice tight bundles we call chromosomes. Also, we can see that in prophase, the nucleus starts to break apart. So when the nucleus breaks apart, this is going to allow for the chromosomes to be able to move through the rest of the stages of mitosis. The next phase of mitosis is called metaphase, M for middle. So what we're going to see in metaphase is that the chromosomes are going to line up in the middle of the cell in a place called the metaphase plate. And so you can see that here in the cell, right? All the chromosomes were kind of, they were condensed in prophase, but now we can see that they're lined up right in the middle of the cell, okay? And so what we're also going to see in metaphase is that we have these two items on the two opposite ends of the cell. These are called centrioles, and they are going to shoot out these things called spindle fibers. And these spindle fibers are going to spread out and then attach themselves onto the chromosomes. And they're actually going to attach to this little middle part of the chromosomes called the centromere. And once those spindle fibers have attached to that centromere, it's going to be able to pull them apart in the next phase. Okay, so now we're in anaphase. Anaphase, A for apart. This is where those spindle fibers that have attached onto the centromeres, the middle portion of those chromosomes, is now going to shorten, right? And so they're going to then start to pull those chromosome pieces to opposite sides of the cell. Now, what do we call those chromosome pieces? We call them sister chromatids, and they are half of a chromosome. So we are going to have essentially, we're pulling the DNA material in the form of sister chromatids to opposite sides of the cell. And we can see it here. The sister chromatids are being pulled to opposite sides of the cell. All right, now we are into telophase. That's we're getting close to the end of mitosis. So at this point, the cell membrane is going to start to pinch in on the side. So you can see the cell membrane is starting to pinch in. We can see that it's looking like the chromosomes are getting pulled closer to these opposite sides of the cells, and it looks like almost two new cells are about to be developed here. Okay, the chromosomes are going to start to bunch together, like we can see, and we need a new nucleus, right? So around those chromosomes, a new nucleus is going to be formed, in each of these cells. We're also going to be able to identify this thing called a cleavage furrow, okay? And that is really you visually seeing the cell membrane starting to pinch in on that cytoplasm. And then finally, we have the last step of mitosis, and that is cytokinesis. If we think about what that word means, cyto means it's referring to the cytoplasm, and it literally means the cytoplasm is splitting in half. And we can see that here as the cytoplasm splits in half. We now have two cells, two identical cells that have been formed. What do we call these identical cells? We call them daughter cells. And now in each daughter cell, we can see each with their own nucleus and their own set of DNA. So a quick wrap up to visualize the cell cycle. Remember, we have two major phases of the cell cycle. The first phase is interphase, which is 90% of the cell cycle. We have G1, where we're going to have the cell grow. We have the S phase, where the DNA is going to copy itself. So there's two sets of DNA. And then we have G2, which is the final growth and finalizing phase to get ready for mitosis, which we have 5 phases. Prophase, which we can see the chromosomes condensing. Metaphase, where we can see the chromosomes lining up in the middle of the cell. Anaphase, where the chromatids, the sister chromatids, are going to start to be pulled to the opposite sides of the cell. Telophase, where we start to see the new nuclei forming in each of these cells. But we still don't have a full division yet, but we do see the cleavage furrow. And then finally, we have cytokinesis, where we see the splitting of the cytoplasm and two identical daughter cells from the original parent cell. Thanks, guys.